Anaerobic respiration is going to be respiration in the absence of oxygen. Now, when we talk about normal cellular respiration, oxygen does play a very big role in helping that process to be very efficient and to produce a lot of ATP. So what we're going to see is that since we do not have oxygen, there will be some different steps involved, and overall, we're going to make much less ATP. In fact, in this case, we're only going to make 2 total ATPs when we start with 1 glucose and we break it down. Other things about anaerobic respiration, this is going to occur in the cytoplasm of the cell. So this will not involve the mitochondria at all. And we also want to say that the end product will be organic. So in other words, we're not going to, in this case, be taking our glucose or our food source and turning it completely into inorganic carbon, which would be carbon dioxide. We're going to be left with organic end products. And just for the 2 different types of fermentation that we're going to go through, one of them will be ethanol. And then when we look at the other one, it's going to be lactate. Regardless of whether there happens to be oxygen present or not, all respiration, aerobic and anaerobic, is going to start in the cytoplasm with glucose. And specifically, it is going to start with glycolysis. So with glycolysis, what happens is the glucose will be converted into 2 pyruvate molecules. And when we have that conversion into 2 pyruvates, we are going to take 2 ADPs and they will get converted into 2 ATPs. At the same time, we're going to have 2 NAD pluses come in, oxidize the glucose, and when they do that, they will turn into 2 NADHs. That's just normal glycolysis. Now, if there is not any oxygen present, what happens next is going to be a little bit different from what we talked about with aerobic respiration or cellular respiration. So we have 2 different methods for anaerobic respiration, or what we call fermentation. So if we talk about the lactic acid fermentation first, lactic acid fermentation is going to remain in the cytoplasm, and it's going to take that pyruvate molecule. And now the pyruvate itself is going to come back here. It's going to accept the electrons from the NADH, or in other words, the pyruvate is going to get reduced. And when it does, it will turn the NADHs back into 2 NAD pluses. And when it does that, it is going to produce 2 lactate molecules. These lactate molecules themselves are both 3 carbon molecules. And recall that the pyruvate itself was 3 carbon molecules. So we start with 6 carbons in our glucose, and we really still have 6 carbons that are organic inside the cell. So if we talk about the overall yield of lactic acid fermentation, it is just going to be the 2 ATPs that we see up here. The lactate, even though it's an organic molecule, in this case, this is considered waste, and that would be the end of the lactic acid fermentation. So what we've accomplished with all of this is that we do produce some ATP from our glucose that we started with. Really, there's just 2 of them, so it is much less efficient than when we have oxygen present and we can run normal cellular respiration. And another thing that's very important with these steps that are shown in blue here is that these are going to regenerate the NAD+. This is an important step because NAD+ is a reactant for glycolysis. And if we do not regenerate the NAD+, then glycolysis itself would stop. And if that were to happen, that would eliminate the possibility of even making those 2 ATPs. Our other type of fermentation that we have is what we call alcohol fermentation. And this one is called alcohol fermentation because the end product, that organic product that we get, it is going to be an alcohol molecule. And so again, this one is going to start with normal glycolysis in the cytoplasm. And what's going to happen to that pyruvate is that it's going to first be converted into two molecules that we call acetaldehyde. Now, when pyruvate is converted into those two molecules of acetaldehyde, we will also be removing two carbon dioxide molecules. So we do, in this case, take two 3-carbon molecules that are called pyruvate, and we convert them into two 2-carbon molecules that are called acetaldehyde. Now what's going to happen is that acetaldehyde itself is going to get reduced, or it is going to receive the electrons back from NADH, which then NADH turns back into NAD+. And the end product over here is going to be that we get two ethanol molecules. Ethanol is a 2-carbon molecule as well. So if we review here our product or our energy yield, it is right here. We just have our 2 ATPs, so similar to what we had with lactic acid fermentation. And also, if you note right here, we did regenerate the NAD+. Other things, we end with a waste product that is an organic molecule, which is the key really when we're talking about fermentation. We're not really completely oxidizing the glucose, because if we did completely oxidize the glucose, we would lose all 6 of those original carbons in the form of carbon dioxide. Let's take a few moments and talk about the differences and the similarities between aerobic respiration and what we call anaerobic respiration. Well, first off, if we talk about what they have in common, both break down organic molecules. And the reason that they both do that is that they both yield energy. Another thing that's in common is that they both start with glycolysis. And if they're both starting with glycolysis, that also says that they both start in the cytoplasm because that would be the location of glycolysis. And then one last thing they have in common is that both require 2 ATPs to get started. Okay, and that would actually be required during that glycolysis part. Now, if we talk about things that they have that are different from each other, if we start off over here with aerobic respiration, first off, this one definitely requires oxygen gas. That one requires oxygen. Anaerobic respiration, no oxygen is going to be required for that. Because of that difference in the oxygen, there's a huge difference in the efficiency. So over here on the aerobic respiration side, we usually say that it can produce a maximum of 38 ATPs, and that would be per glucose that we start with. And on the anaerobic side, it's going to be just 2 ATPs. And these are ATP profits that we're talking about. If we talk about other molecules, as far as end products go, aerobic respiration is going to release all of that original glucose molecule in the form of carbon dioxide. So we would exhale that. On the aerobic respiration side, we do have the 2 types of fermentation. If it's lactic acid fermentation, lactate would be the waste product at the end. If it's the other type of respiration, so ethanol fermentation, then we're going to have ethanol and some carbon dioxide produced as a waste product or an end product.